Hello and welcome to Module 9. This week I decided that it was time to deviate a little bit from the syllabus so that we could provide you with more tools as we reach deeper into just dissecting the scientific literature. You're in your second journal club now. The third and final journal club is going to start soon, and then you'll be on to your final project. So it's really important, I think, at this stage that we take a step back and we think about some of the principles guiding the literature that you're becoming more familiarized with, and that you are very closely working with each day in this course. So let's talk a little bit about how this data is actually arrived at, how we are able to generate the tables that we need in order to do these kinds of studies. If we start with a FASTQ file, how do we then move forward into something that we can create a story out of and then a scientific paper? Right. Okay, so hopefully this is a welcome deviation from our tentative plan for the semester. Next week we'll go back into the 8 microbial niches. We still have ocean, animal, and as much of plants as we can get done, although a lot of that was covered in soil before we wrap up the niches, that is. So, okay, let's go ahead and get started on the materials that I have prepared for you here. Okay, so the more practical aspects of microbiome research and data sets. When we think about how much data there are, it can be a little bit daunting. Where are we going to house all of this data? How are we going to work with the kind of memory constraints that are necessary to make progress with these big data sets? How do we make questions out of this material? Okay, so for the outline, we'll go over a little bit of the compute process, data set constraints and considerations, processing considerations, and then finally our scientific question. And I do want to point out that here in the module, there are several resources that have been provided for you as required reading. For this one, actually, I don't want you to read very much of it. I only want you to focus on the specific sections that I've outlined. You're going to be skipping a little more than half of the article. For this one, all I want you to do is focus on figure 1 and table 3. You're welcome to read the rest of the article if it interests you and if it's important to you, but I want you to focus on figure 3 here, and also figure 1 here, and then table 3. Okay? Finally, there's a data 2 tutorial. You are not being asked to run any of this code yourself. I do not want you to run this code as part of this class. I do not want you to have to work through any of those details, but what I want you to be aware of is what happens when we have a 16S data set or when we have a long read sequencing data set. This is not specific to metagenomics. That's a much more complicated bioinformatic processing pipeline. We're not going to get into that. It's beyond the scope of this class, how we arrive at the final processed data that we would analyze. But I did think it was important for you to be aware of data 2 for 16S applications. So what I'd like you to do is just read through this. There's a very nice annotation as well as example figures. So put yourself in the shoes of someone who would be running this in RStudio. That's what we're running it in, is RStudio. It looks like this, RStudio. And you can see that memory can be exhausted. It's definitely a constraint when working with these large data. So if I were to follow along on the tutorial with my own data set, then I would eventually arrive at let's see, let's scroll through these steps, which you should outline, kind of be familiar with the process, what comes first, what comes next, and why is it important. Here, once everything is done, you would then hand off to what is called PhyloSeq. So PhyloSeq is a package in R that basically allows us to create a single object with three parts. The first part is how we've assigned each amplicon sequence variant or operational taxonomic unit. So what does each of these sequences align back to? Which bacterium? So it's basically a table showing each of those bacteria and what they are. So that's called your taxonomy table. Then there is the OTU table, which has for each of those sequences, each of those IDs, the counts. So how many times is this particular sequence seen in a given sample, right? And then the third is information back to the samples themselves. So any metadata, any characteristics, any features that we want to capture about the samples, like collection site or age or sex or some kind of, whether they're on antibiotics or they have a certain color of hair, whatever the trait is, those features would go into the sample data. So again, there's tax table, OTU table, and sample data. So what we're doing here is taking what we've created through data 2 and loading it into a phyloseq object. We're creating a phyloseq object right here, this one line from this variable or from this object rather. So the OTU table would be this that we just created in data 2. The sample data is like a CSV or an Excel file, just a table that we're feeding into the program, telling them the features, and then the tax table is the breakdown of the taxonomy. Okay, so then when we type in the name of the phyloseq object, we get information. We know immediately, okay, there are 19 samples here and 232 different bacteria across those 19 samples. Doesn't mean that every bacterium is in every sample, but it means that bacterium was seen at least once in at least one of those samples. We only have 4 variables that we're mapping back to in these samples, and we have the 7 taxonomic ranks. So when that tax table is called in, we actually see kingdom, phylum, class, order, family, genus, species, and we can look either at each ASV or each OTU, depending on how it's defined, or we can look at a particular species or a particular genus, etc. And then at this point, it's very easy to do things like visualizing diversity. We'll go over that in this lecture as well, at least just making sure you're familiar with different metrics for diversity. We can do all sorts of beautiful plots here, a bar plot of the relative abundance of each of these families, for instance. So as I mentioned, we can tell R that we want to fill in this graph with family level conglomerated data, right? So we're going to condense everything into family rather than having, hundreds of different colors here for each ASV. Okay, so now if we make our way backwards, um, now, if we make our way backwards, so this is the starting point. Be sure that you're aware of what the assumptions are, what has already been done before we get to this point. And again, this is all to familiarize you with what really happens after we sequence samples for 16S. We've seen this done in the studies. What do they do? How do they get to these answers? Well, they have to demultiplex or break apart the sequences based on which sample has which sequence. Non-biological nucleotides have to be removed. And then the forward and reverse FASTQ files, so you have your forward, your reverse, they have to have reads in matched order. Okay, so once that is taken care of, we've gotten rid of primers, that kind of thing, then we can load data 2 as a package in R and do each of these things. Data 2 is specific to R, so this has to be done in that program. Again, RStudio is fantastic as the interface for R so that you can see everything that's going on. Each of these steps, it takes a different amount of time. It really depends on how much data you have. So that is why I noted here that there's a slightly different version of this when there are big data sets. Um, the reason why is if you try to run a couple of these steps, if you try to run one of these steps that takes a lot of memory, you'll heat up your computer really quickly. You won't know what's going on unless you say, you force verbosity. So you're forcing the program to write some stuff out, how far you've come in your progress. Your computer might overheat, and then you're going to be told there's an error, vector memory was limit was reached. So this allows us a workaround where we basically work with the data in chunks. So we take care of this chunk, and then when it's done, we move on to this chunk, etc. And then it's more manageable, and it won't fail, right? So it's really important to keep in mind these considerations. This one step alone can take 2 to 12 hours. And this is part of the reason why, for an introductory course such as ours, we do not expect, nor do we want you to run any of these things yourself at this stage. We just want you to be aware of what happens. Okay. So, again, this process is a bit different for metagenomic sequencing. There are a lot more players at stake. There are a lot of other packages that have to be used, but this is one approach if we have 16S or ITS sequencing. Okay, so that's why you'll see this requirement here. Okay, so some basic things that have to be done. Uh, uh, we have to filter, we have to trim the sequences based on quality. That's very important. Data too can help us to accomplish a lot of these points. So we look at the quality, we decide where we need to trim things because we start to lose quality. Uh, we can also learn the error rates. So every data set has a different set error rate, and this is something that we can then take into account in the processing. We can visualize the estimated error rates and just make sure that things look appropriate before we move into inference, right? And trying to infer what sequences are actually there in each sample. If the paired reads haven't been merged at this point, then they'll be merged, uh, and we'll construct the table. Okay, so the other consideration here is we have to assign taxonomy. So once we have sequences, that's not enough. We have to then match back to living databases that help us understand, okay, well, what exactly is this sequence? So you'll see here in the data to pipeline, there are several options for how to arrive at taxonomy. And this is another area where we can introduce, bias or constraints. So we just want to be very clear with ourselves and with others what database we use for training, what database we use to assign these taxa. So there's the RDP, the green genes, and the Silva. The Silva is what I like to use for these applications. And there's a little bit of a process for downloading these data sets. So you just want to make sure that you do get them downloaded and that you can then use that for assigning your taxonomy. So if you were to go to the database to download the files, that's one option. Um, so to um, we can go to the Silva website to download these databases. Creates uh, like a um, it creates a cart for us to download them. Um, but I actually went straight to Zenodo to download mine um, most recently. So here in Zenodo, the training data has been put together in these files. That's the easiest way for me to get to it. So there's an add-on if you want to go to species level, and then there's genus level and assigning species. So you would download each of these files. They take up a little bit of space, and that's what you would call into data to. Um, so that is the step. Where is it? Um, That is the step here where we're assigning taxonomy. So what you would do here, again, we don't expect you and we don't want you to do these things right now. We're just making you aware, is you would fill in this line with the exact location on your computer or on your cloud where those files that I just showed you have been downloaded to. Okay? So then R knows where to look for them. Okay, so again, these are very practical aspects of the microbiome. These are things that you should be aware of. How do we go from here's the sequencer to, okay, here's a data set that I can work with? All right, so now that we've covered that, and once you have a phyloseq object, there's so much that you can do with it. Some other considerations for your data set, how you collect and store the sample does make a difference. That shouldn't be a surprise to anyone here. There are kits that are available commercially for preserving DNA or DNA and RNA. They work very well. Like Zymo or Isohelix, for instance, have been used for many, many years. So these are considerations that you have to take into account when you're designing your study. How long is the DNA or the DNA RNA, how long is that going to be sitting around for? You want to make sure that you take into account, well, where am I going to be? Am I going to be in the field where I can't carry around dry ice? Am I sending out kits where I'm not sure if somebody's going to collect them and put them in their freezer right away at their home? What are some of the constraints of the clinics that I'm working with? Do they have access to freezers to house this until they can send it to me, right? So there are a lot of considerations. Freezing at -80 is considered a gold standard if you have the sample itself, but sometimes that's not necessary. If we just want the DNA, RNA, we can purchase and build up our inventory of these kits that have proprietary enzymes to preserve the RNA, DNA, or preserve just the DNA, and then we can move forward. The disadvantage there, of course, is then there are things that we can't look upon because we are preserving just those components. We can't look at like metabolites, for instance. But again, it's just a question of do you really need that information downstream in your study? If you do, maybe you want a combination of these. Um, okay, and another thing I wanted to bring up to you is this concept of eDNA. So that can be very useful where we could sample a certain environment, sequence it, and get back a lot of information about what's in that environment. So not, just enriching for a specific species, a specific, we're looking at what is in a certain person or in a certain animal or in a certain sample, but we're looking at all of the environmental DNA. We can kind of trace back everything that has been there in a certain period of time. So it's kind of interesting application for genomics, for just creating a structure, a history of who has been where, when. That I just wanted to make some of you aware of in case it's something that is important to you and the questions that you have. Extraction methods can also dramatically affect results, so you want to standardize this as much as possible. Find something that works, optimize it, stick with it. So mechanical disruption techniques, there are some things that can shear the DNA and make fragmented, make kind of a fragmented mess, things that you don't necessarily want. One of the key principles is garbage in, garbage out. So if we have garbage in our data set and we're training on that and we're working with it, we're going to get garbage results, right? So these are aspects that I don't want to underplay at all. They take a lot of intentionality and foresight and just thinking things through. So for sequencing and processing, we have talked about this a bit, different platforms that are available. I think, something that you should all keep in mind is that metagenomic sequencing is actually quite powerful because we can look at the genomic content, not just what bacteria are there, but what they're capable of doing. And I don't know that we really drove home that point before. One of the issues, of course, is the computational opportunities that come with such a data set. It's enormous. So if you're just, amplifying a certain gene and this is what you're getting, that's one thing. But if you're trying to go to metagenomics, it's a much, much, much bigger data set. You do want to take into account possibly commercial communities, right? So having a sample where you know what's in it because it's been commercially made available to you, or having blank controls, and being careful with what you're trimming. You don't want to be overly conservative and wasteful and taking out too much data, but you also want to make sure that the data that you keep when you're making decisions like where you're going to trim for quality are justified. Um, I wanted to bring up yet again these memory requirements for the bioinformatics pipelines. Luckily, at University of Florida, we do have the NVIDIA machine, which is great. But just things that we don't always consider when we go into a venture like this. We're going to have issues with how we can process these data, and we need to think about that and be smart about it. There are options such as cloud services that can be used for housing these files, appropriately handling them, no mishandling of data, classified information, especially if it's health information, making sure that you're in line with your IRB. There are a lot of ethical considerations that need to be taken into account. Um, and even with some environmental sampling, we have to be very careful and make sure that we're doing our due diligence. But I did want to keep that on your radar that there are cloud services and things that we can leverage so that we're not eating up everything on our local machine. That's not always the best idea. Okay, so now on to the compute, which, as I've stated time and time again, is really important to take into account. So let's say you have 500 samples in your study. If there are 10 million reads a sample, because we're doing metagenomics, that compressed file can be 1.2 to 2 terabytes. That's a lot, right? Some computers don't even have that much storage. Okay, that's just for one study. For metagenomics, these large data sets, the raw reads, what we get off of the machines can be 2 to 10 gigabytes per sample when they're zipped, when they're compressed. On top of that, you have to plan for at least 2 or 3 times the size of your raw data so that you can handle all the intermediate steps. Again, what we went over here in the data to pipeline is just a small piece of what is necessary if we have metagenomics, right? We have to take that into account. We have to make sure there's room, or we're going to get very frustrated because we're not going to be able to move forward. Um, so you have to plan for at least 2 or 3 times the size of your raw data so you can handle these intermediates, the trimmed reads, the contigs, the annotations. And then memory, our dear friend memory, our RAM, assembly-based pipelines, which we would use in metagenomics, may require as much as 130 gigabytes of RAM for large samples. Okay, so these are considerations that you have to take into account when designing your study and when thinking about who your partners are going to be, what you're going to do with the data, how you're going to handle this. Um, don't underestimate it. Okay, so I want to take some time now to talk about something a lot of people don't dwell on, and that is your model system. Okay, so if we have a question in mind, what is the appropriate population for that question? Who do we study? Sometimes it might just be who do we have access to, or whom do we have access to, right? Sometimes it might be an issue of um of of I don't know um, who may have interest in the study, where we are geographically, uh, what populations of people might be willing to do the things that are in our IRB for like an intervention study with diet or lifestyle, let's say. But something that I think is really key to keep in mind is the translatability. So, like with animal models, for instance, I have an inherent bias. I always go to human models, always. And there are a lot of reasons for that. I'm happy to unpack with any of you at any time. Just send me an invite and we can talk about it over Zoom or something. But um, a lot of models are not necessarily translational to real life human applications. Okay, even studying a human in a lab, it may not be translatable to a human in the real world, right? So there are a lot of environmental factors at play. So I urge you to consider, are the decisions that I'm making really worth it? Are they translatable to the question that I have? And along those lines, what is the inherent expense? So there's always going to be data that's wasted, time that's wasted, sample that's wasted. How can you minimize that waste and be as efficient and effective as possible? Um, so, for instance, in some animal studies that require a certain genetic background, what happens to those who are not in that genetic background? Right. So I'll leave that to you to fill in the blank. Um, so these are things that aren't really harped on at the end in the story in the paper, but they're very real considerations that we have to make. Okay. And part of my role is to remind you of those considerations so that you're equipped when you're leading your own study or you're invited to take part in a study to bring up these points, right? Because not everybody in the room will. Okay. Then for pre-processing, there are obviously memory requirements planned for intermediate files. We talked about these statistics, these numbers, this arithmetic earlier. Big data means big storage, and where can you house it? And there are ethical considerations in cloud services. Make sure you pay attention to access and security. Um, so let me see, where are we in the full sequence? Okay, got it. So in the processing, the reason I have a picture of this beautiful plate of food here is because for me, this reminds me a lot of how we would work together in a kitchen. Um, we have to make decisions about how we would standardize our recipes, how we would standardize how we deal with these reads. Um, there are a lot of pieces that come into account, especially with rarefaction. So my grandmother grew up in the Depression, and because of that, she was very in tune with waste. And I wondered sometimes why she always ate everything on her plate. And then when I learned of her background and her history, it made so much sense to me. She grew up in a world where there was very little. And so everything that was on that plate was going to be eaten. Nothing was wasted. And this is how she is decades later, right? Um, so that's why I thought, it was important to bring up waste not, want not. Why rarefying microbiome data is inadmissible. It's a very controversial kind of paper. This would be a perfect example for a TED Talk, right? Um, so I forgot to put this in your required reading, but I will figure out which aspects of this paper I want you to key into and to read. But I at least want you to be aware of this paper. It's very important. It's been cited a lot of times. It's one that I go back to a lot when I'm making decisions. Obviously, when we're sequencing these samples, we're going to have some uneven sequencing depths. Some samples may have more reads than others. Some samples may be more successful than others. So how are we going to account for that? Right? Especially when we have issues like diversity, where we're looking at how many different kinds of species are in a sample, let's say. Well, if you have more data, then you have more opportunity to have different species, right? So, uh, so we can one option is to rarefy, but the downside of that is that we reduce our power, our statistical power, because we discard data, especially from samples where we have very high sequencing depth. There can be artificial variation and bias that we introduce because of that random subsampling. What if we're not taking the right piece of that file, right? So let's say that we cook something a little bit unevenly, or we have a plate of food, and this side has a lot of ingredient A, and this side has a lot of ingredient B. And it just so happens if we sample the plate from the left, we're going to get a lot of B. If we sample it from the middle, we'll get both. And if we sample it from the right, we'll get A. But actually, that doesn't that little subsample isn't representative of the whole. So that's something to be considerate of. And depending on where you set your sequencing depth when you're rarefying, let's say you have a few samples that didn't reach that depth. That means you can't sample that much, right? So actually those samples get discarded and we waste them, right? But if we go too low, then we waste a lot of data in other samples. So let's say we set our threshold at 50,000 and we have samples that are at 30 and 10 and 40,000, all those samples would get thrown out. We wouldn't analyze them. But if our other samples are 50 to 150,000, we don't want to lose so many reads. So these are a lot of important decisions that have to be made. So it has been studied that doing rarefaction repeatedly, iteratively, can help us to assess the impact of removing the data and improving power. That could be an option. There are also options like scaling, different scaling methods. So cumulative sum scaling or variance stabilizing or upper quartile, where you're normalizing based on a certain quantile of reads. Transformation, which is an approach I like to use when we transform relative abundances into a new space. So now they're more suitable for analysis. So we can do center log ratio transformation, CLR. You might be thinking, oh my gosh, this sounds way too complicated. But guess what? Once we have our phyloseq object, as here, we can do any of these things. It's actually just a simple line of code. And we say what transformation we want to make. Obviously, since microbiome data are compositional, so meaning that they all add up in a sample to 1 or to 100%, that's something important to know, compositional nature of microbiome data. Everything is relative to everything else in the sample. So we could look at like relative abundance, for instance. So if we look at everything in the sample, we have 50% bifidobacterium, let's say, or 20% of this other species, right? Okay, so that's a consideration. A lot of the statistical approaches that you can take require just the count data. So the raw counts, the integers, the zeros, the ones, the twos, all of that, the 300,000. And once we apply data scaling or transformation or rarefaction, we no longer have valid count data. So you just want to make sure you have your count data around. Okay, there are a lot of other nuances to that that we didn't go over, but I did want to mention this little tool at the top. It's imperfect, but it's a start. It's free, and they have some very simple, small data sets you can play with. Microbiome Analyst. What you could do is load in one of their free open source data sets that are quite small. Everything's online. You don't have to download it. And you can see how applying different rarefaction, scaling, transformation approaches changes or doesn't change the end result, the end conclusion, the statistical results from those data sets, right? So you could apply the CLR transformation and then run a couple tests and see what happens. You could apply rarefaction, etc. Okay, so then on to your scientific question. All right, we've talked about the model system, asking yourself, is the population that I've chosen the appropriate choice for my question? That may evolve with time. What inherent biases are introduced with those decisions? Confounds are very important. I was working with a data set just last week where once I adjusted for a certain confound in diet, I got wildly different results than when I hadn't adjusted for diet. And things that I was missing before, associations with a periodontal pathogen actually became clear, right? So once I accounted for diet, once I accounted for this question pertaining to how much sugar somebody has every day, then the association with the periodontal pathogen came through because it was so strongly linked to the sugars. So once I accounted for the sugars, now this association came up through the noise and I could see it. So confounds are really important, and there are many studies where they just say, "Oh, well, I adjusted for sex and age." Like, okay, well, is that enough? I don't know. We can't adjust for everything, but what is practical and what is necessary to adjust for, or at least explore in our data set. So when we talk about community level and multiomics, you might want to consider, are there other data that could support your hypothesis, right? Like genomic data or metabolomic data, proteomic data, transcriptomic data, right? All these other kinds of things. And how could you use that in your study? We'll talk about diversity here in a couple minutes. And I want to drive home the point that no single path is best. Um, you should embrace that. Okay, so sometimes um, you have to allow the opportunity and the moment to guide you into which direction you need to take with your data. Okay? Simple. Keep it simple. Okay, so microbial transmission. How are microbes moving into a host? Well, they can move vertically, top down, let's say, like vertical transmission, inheriting them from the parent, or horizontally, which is probably what we're more familiar with when they're acquired through the environment, like food or sharing space with individuals of another species or the same species, right? So how we interact with our beloved companions, our dogs or our cats, how we interact with each other. So horizontal transmission is where we focus a lot of our time, but there's also vertical transmission, what we inherit. So then for diversity, we can define diversity within a sample, which is alpha diversity, or between samples, which is beta diversity. There's also gamma diversity, which is the diversity across all samples. So within alpha diversity, it's important for you to know that that can be defined by two different things. We can look at either the richness, so that's how many different species are there, how rich is it, or by the evenness, which is how evenly are the species that are there distributed, right? So very, very different. How many species are there? And then how evenly are the species distributed? Because you may see 1,000 different species, but if two or three species are really driving the community, it'd be very rich, but it would not be even, right? Or let's say there are only 20 species present, but each of those is has like an equal say in the community, then it's not very rich, but it's very even, right? So these are some considerations, ways you can think about this the sample. So for richness, observed species in Chow 1 are indicators of that, and then the Shannon and Simpson index are examples of evenness. So when you see these terms, now you'll know, okay, we're talking about how many species are there, or we're talking about, irrespective of how many species are there, how well distributed are they? So in that case, a community with 10 species that are equally abundant would be more diverse, right? Than a community that maybe has 100 species, but it's dominated by one. Okay. Um, okay, so Shannon is often used, but I did want you to be aware of these others and that they provide complementary information, right? And we can have different results based on them. So what you would do here in a study is look at the alpha diversity according to, the different definitions in each and every sample. Then what you could do is compare the alpha diversity seen in samples in group A to the alpha diversity seen in samples in group B, right? And then you see if that's statistically different. Okay, then the beta between samples is capturing how different two or more microbiome communities are. But between the samples. So for abundance, Bray Curtis is used, presence absence, Jacquard, and then for phylogenetic relatedness, when we're taking that into account, Unifrac is what is used. So now you should have a basic understanding of what each of these terms means when you see them. Okay, so I did ask you to read various portions of this paper. It's kind of a long paper, so I don't want you to read the whole thing. I've written out every single section that I want you to read, and this section's kind of long, so just the first, second, and the last paragraph of that section. Um, but the holobiont is basically um, the summation of a host, however the host is defined, and all of the microbes associated with that host. Okay? Hologenome would be the genome of the host and the microbes. So every host has a core microbiome, and those are present in most individuals in that species or most people, right? They carry out very essential functions. Okay. So that's a core microbiome. And then there's a peripheral microbiome, core peripheral, where there are all these other taxa that carry out other functions. Um, so even if there are very big differences in someone's environment or in their, just their lived experience, um, uh, some microbial communities may vary a lot less. Well, yeah, microbial communities will vary a lot less among individuals within a species than between different species. Although it is interesting because dogs and humans, their human companions and canine companions share a lot of their microbiome, which is quite interesting. So this is something that people are curious, very curious about how much of our biome is dictated by just our environment, even if we're a different species, and how much is dictated by just who we are, right? But the reason I brought this up is because of this concept of a meta community. So if we're going to study, let's say, human drivers associated with a certain outcome, right? So we're looking at microbiome drivers of a certain outcome. Um, there's also environmental drivers, right? So all of these kind of intersect with each other. It becomes very complex, very complicated very quickly. This is what the authors describe as the human environmental microbiome nexus. So it's a complex, functionally important network of human and population and in the context of a trait or disease or health. So as I mentioned before, confounds matter, environment matters. So just kind of appreciating the interconnectedness of all of this is important, and for you to be aware of when you're reading the literature critically, and also when you're thinking about your own studies. All right, so we've covered a lot today. Um, and what I want to leave you with is that not all questions lead to a clear answer. Not all questions worth asking in life lead to a clear answer that is always the same, right? So as we evolve, as the field evolves, as questions evolve, um, the answer or the path to that question may change, and that's okay, right? So I think as long as we're aware of the biases, as long as we're aware of the decisions that we have to make, how we justify these decisions, we can move forward with the best information that we can have at that moment in time. And that's what we owe to ourselves. That's what we owe to the science and to others, is to lead with the best information we can, make our decisions, and move forward. But it might mean that we have to go back to that sometime with more information, with more data because this picture is constantly evolving as we're defining ourselves, as we're defining the question. Okay. And that's all. So I want to thank you for your time. Thank you for your attention. And hopefully you found that this was an interesting consideration with some practical applications as you move forward in the course and move forward in your understanding of microbiome data and microbiome studies. Thank you, everyone, and take care.